What do you do with your hydrogen ions? Increase secretion. So what should the urine pH be? Acid. Yeah, urine pH should be acid, because if you increase secretion of hydrogen ions in your urine, that includes excess hydrogens, means your urine's going to be acidic. So that's one way to actually determine that a person's, compensating for a respiratory issue. This person, are they currently hypo or hyperventilating?
If they're right there, they're hypo.
Yes, ignore this right now. Just looking at these numbers. Hypo. They're breathing too slow. They're not getting enough CO2 out of their bloodstream. So just based on this problem here, you know that this problem was caused by respiratory acidosis. That could be caused by hypoventilation. This could be emphysema. This could be COPD. This could be diabetic acidosis. So you've got a number of things that could send you there.
Did you say what the normal range was for bicarbonate?
Bicarbonate is 22 to 26.
So that would have been a blood pH of 7.3, and then something with the bicarbonate, you knew it would have been metabolic because of the...
Yeah, if you had, give you one over here. You're saying 7.3, which is acid. That's considered acidic in terms of bicarb. So now those two match. Now you've got
metabolic acidosis.
Bingo.
You'll never 137. PCO2 normally is 35 to 45. So let's say that you've got a PCO2 right now that is 20. Okay. So is there any compensation right now?
Yeah, because that's basic.
Yes. There is compensation. We're not in the normal range. It is, in fact, basic or alkaline. So the compensation here is respiratory. There you go. And what is this person doing? Hyperventilating. Yeah, hyperventilating is going to help solve this problem. So what you want to feel comfortable with on Thursday's test? I can give you some numbers. I might ask you to diagnose the problem. I might say to you, we've got somebody who's hypo or hyperventilating. You'll want to say, okay, that leads me into acidosis or alkalosis. You also want to feel comfortable providing the compensation. The compensation is not just saying, okay, you've got respiratory alkalosis or metabolic acidosis, but take it the next step and decide, is the person hyperventilating? Are they increasing, decreasing reabsorption of bicarb? And are they increasing, decreasing secretion of hydrogen ions? So it's not just memorizing how to do the problem, but getting the holistic picture here in terms of what could cause the imbalance, and then how do you compensate for that imbalance.
The last thing you said was increasing or decreasing
Hydrogen secretion, right. How about other questions with acid base? Is it starting to click for you? Are we still in mud right now? Okay. Why don't I go ahead and get you into the reproductive system then, and do you need to hear all four of these topics? Okay. We'll see if we can do a speed demon through all four of them in the next 45 minutes to an hour. You probably have heard of mitosis in Bio 210. That's the topic we commonly cover. Meiosis, or meiosis, however you like to say it, is probably a less commonly covered topic. And the thing to remember here is that in sperm cell and egg cell production, we are looking at using the process of meiosis to make those sex cells. I'm going to let you read about all the anatomy of the reproductive organs. I figure that's something you're probably familiar with from lab. So I thought I would focus with you here in your course pack on page 144 for spermatogenesis. And as soon as we finish that, we'll talk about oogenesis over on page 150. Okay, so in your textbook, they talk about spermatogenesis on page 903. The process of making sperm in males is called spermatogenesis. This process occurs at puberty and continues until the day the man dies. That's a long time to be making sperm. Women undergo a process known as oogenesis. Oogenesis partly begins before birth. It kicks into full gear at puberty. But of course, women go through menopause, and that stops sometimes in the 40s or 50s. So a woman has approximately a 40-year cycle over which she can produce those eggs. So women don't produce eggs until the day they die, unless that's sometime in their 40s or 50s. That process does come to a stop. Both spermatogenesis and oogenesis rely on this process here called meiosis. So I wanted to walk you through meiosis first before you see spermatogenesis and oogenesis. This is a great website. If you have trouble going home and getting this stuff straight, I would encourage you to look at this website. It's on pbs.org. I'll post a link on D2L for you, and it's just a real nice, simple animation that shows mitosis versus meiosis. Alright, so they're going to start you off here with interphase. And in interphase, late interphase, we're going to start forming those mitotic spindle fibers using the centrioles. Okay, and the chromosomes are actually formed here by the time you reach full-fledged in the prophase. So we know that during prophase of both mitosis and meiosis, we've got chromosomes present, we're starting to build the mitotic spindle fibers. What are we setting up to do during mitosis?
Duplicate the cell.
Duplicate the cell, exactly. Does the daughter cell that results from mitosis resemble in any way the parent cell? Absolutely identical, right. In the case of meiosis, however, what we're trying to do is duplicate that cell, but ultimately we're ending up with 4 total cells, and the daughter cells do not resemble the parent cells genetically. In fact, the 4 daughter cells that are produced are actually genetically different from the parent cells. That's how every single egg that's produced or every single sperm that's made is actually different from the parent cell. That's how you can have brothers and sisters that resemble each other not at all, or they can actually resemble each other closely, depending on how the mix of genes comes apart during this process. Alright, so far everything looks somewhat similar. You might notice here that we have the mitotic spindle fibers set up. Those fibers are going to help guide the separation of chromosomes as they enter into anaphase. You might notice here on the meiosis side, they actually call this prophase 1, anaphase 1. We go through telophase 1, and then we go through a second reproductive cycle of meiosis. It's called prophase 2, metaphase 2, anaphase 2, and telophase 2. So there's actually a second reproductive cycle here in meiosis that we do not have here in mitosis. How many chromosomes does the typical human being have? 46. Those are essentially 23 identical twins. They're 23 pairs of the same information. I want you to notice something real important here. On the mitosis side, all 46 of those chromosomes line up, laying directly across that metaphase plate. But take a look over here during metaphase of meiosis. Those chromosomes have paired up with their twin or their partner. We call them homologous chromosomes because they have an identical twin that carries the same exact information. So what you've done, instead of lining up all 46 chromosomes directly across that metaphase plate, you line up the 23 partners. 23 on one side, 23 on the other side. So what you might expect to happen, as we go through anaphase, you might recall from mitosis, the sister chromatids are separated into separate cells. But look here, we've got entire X-shaped chromosomes being separated during meiosis, so we have not yet separated those chromosomes into sister chromatids. Thanks, Julie. All right, so there you go. Mitosis is done.